AMALearning, you're about to. Specification point 3.8b. Describe the stages of protein synthesis, including transcription and translation. RNA polymerase binds to non-coding DNA located in front of a gene. RNA polymerase produces a complementary mRNA strand from the coding DNA of the gene. The attachment of the mRNA to the ribosome. The coding by triplets of bases, codons, in the mRNA for specific amino acids. The transfer of amino acids to the ribosome by tRNA. The linking of amino acids to form polypeptides. This is a biology-only specification point. To successfully answer any GCSE questions linked to this topic, you will need to know the transcription and translation process in a lot of detail. Protein synthesis is the process by which cells produce proteins based on the genetic instructions in DNA. It occurs in two main stages: transcription and translation. Stage 1 is transcription. This is when DNA unzips around the gene. The DNA strand unwinds around the gene that codes for a particular protein. The enzyme RNA polymerase binds to the non-coding DNA located in front of the gene. The RNA polymerase uses the coding strand of DNA to make a complementary strand of mRNA. Messenger RNA. This mRNA strand carries the genetic instructions out of the nucleus to the ribosomes, where proteins are synthesized. In simpler terms, RNA polymerase is an enzyme that works like a copy machine for making RNA. Its main job is to read the DNA's instructions and create a matching mRNA strand. This mRNA strand is like a recipe card that the cell uses to make proteins, which are essential for the cell to function. So, RNA polymerase helps turn DNA's information into a usable format, messenger RNA. Translation is like building a protein using instructions from the mRNA strand, like a recipe. Here's how it works. The mRNA, messenger RNA, made in the previous step attaches to a ribosome. This is the cell's protein factory. The ribosome reads the mRNA instructions in triplets of bases called codons, which tell it which building blocks, amino acids, to use. tRNA, transfer RNA molecules, act like delivery trucks, bringing the right amino acids to the ribosome based on the triplets of bases, or codons. The ribosome links these amino acids together in the exact order specified by the mRNA, forming a polypeptide chain, a long chain of amino acids. Once the chain is complete, it folds into its unique shape to become a working protein that the cell can use. In simple terms, translation is like following a recipe to assemble the right ingredients, amino acids, in the correct order to cook up a protein. Protein synthesis is a vital biological process that converts genetic information from DNA into functional proteins, which are essential for life. The exact order of amino acids in a protein determines its unique structure and function, emphasizing the specificity of proteins. These proteins play diverse roles in the body, acting as enzymes to speed up chemical reactions, hormones to regulate various functions, antibodies to defend the body from pathogens, or structural proteins that maintain the integrity of cells. However, errors in the transcription or translation stages of protein synthesis, or mutations in the DNA itself, can result in dysfunctional proteins. Such errors can disrupt cellular functions and potentially lead to diseases. Overall, protein synthesis underscores the importance of our genetic code in shaping the proteins that are fundamental to life processes. Let's apply what we have learned to a past paper question. In this question, transcription has already occurred, and hence, it is asking to describe the process of translation. We know from our learning that to make viral proteins, the host cell will need to carry out the process of translation. The mRNA, messenger RNA, made by host cell attaches to a ribosome. The ribosome reads the mRNA instructions in triplets of bases called codons, which tell it which amino acids to use. tRNA, transfer RNA molecules, bring the right amino acids to the ribosome based on the triplets of bases, or codons. The ribosome links these amino acids together in the exact order specified by the mRNA, forming a polypeptide chain, a long chain of amino acids. Once the chain is complete, it folds into its unique shape to become a working protein, which in this case is the virus. If we compare our learning to the mark scheme, we would have got the marks outlined in every single bullet point. So full marks. Well done. Our animations were designed by experienced UK-trained teachers. We have been achieving amongst the best results in the country for many years, and we decided it was time to share our success. Have you just AMALed and want to AMAL some more? Don't forget to like, subscribe and share to keep yourself up to date with the latest uploads. AMALearning, you just did.